Okay, perfect. So I'm really excited to be able to talk at the conference I helped organize. It's such a pleasure to be in a room with many FAIR like-minded people. So let me put my screen here. So the talk I'm giving is really on two things. One, CDD Annotator, which falls into that community tool that could help end users take advantage of ontologies, as well as the involvement of that annotator tool in the DataFAIRy project, which was sponsored through the Pistoia Alliance. So many of you may already know us, Collaborative Drug Discovery, CDD. We started in 2004 out of San Francisco, and we provide a cloud-based data management and discovery platform to the scientific community. And we really have three products. So CDD Vault is our flagship product. It's our most established product, and it's about handling your biologic and chemistry drug discovery data. We have two other products from our semantic suite. And one of the most interesting and exciting things about CDD is that we're really all scientists at heart. And so we have a very active and innovative research informatics team who are allowed to kind of come up with creative new ideas and implement those ideas. And sometimes those ideas turn into something that we can make into an actual product. So our semantic suite, both products, the BioHarmony Semantic Drug Data Store and the annotator that I'll talk about today, are both out of those research informatics teams. And the other products, Whitney is going to talk about tomorrow in his presentation. So all three of these products, so they follow the same core principles. They have to be easy to use, they have to be secure, and they have to promote collaboration. And in the end, we see this as a formula for creating a smart software that saves you time. But one of the things to think about is inside the lab, how do you practically save time? And the answer is you have to be a bit of a soothsayer. You have to kind of try to future-proof your data, right? You don't know if we are collecting and writing down all the important information that we're going to need to answer tomorrow's questions. So we do a really good job with our assays, recording that numeric data and some critical assay description points, maybe temperature, incubation. But what we find is, a couple of years from now, maybe we realize that the pH or the color was important. And because we didn't write it down, we now don't have that data. We can't aggregate our assays as we hoped. And so part of the goal with CDD's products is to avoid problems like this, to maybe avoid the oof moment of I described my assays poorly, and now I don't have the data I need or the time I once had and the answers I wanted. We get so much information now, whether it's from ELNs or SOPs in the house, or even detection instruments, that we just need a really good way to capture it. But more importantly, we need a way to capture it that's easy for the end user, or else it won't happen. So we feel like with one of the products that we created, Annotator, we've cracked that code. And Annotator used to be known as BioAssay Express. We're going through a bit of rebranding at the moment, so you may hear it called a few different things. But it's a system that takes unstructured human text protocols that are in your ELN, in your in-house library, Post-it notes, back of the glove, and really turns it into something that is now machine operable. It structures it using a guided template that is linked to public ontologies, and it's powered by a machine learning natural language processing engine. And it works in harmony with the human. So these ML and NLP steps are meant to speed the curation process, but they don't remove the human from the loop, because what we're interested in is getting really high quality and really accurate metadata. And that idea is once you have that really accurate metadata, it becomes just another axis or another point for aggregation or detail that you can use in your bio or chemical analyses. We do it the other way too. So we can also take assays that aren't written yet. So on the fly assay generation using that form, and then spit it back out into something that looks human readable, even though it's derived from a machine readable format. And that sometimes is easier for scientists to deal with, at least on a visual level. And then that also can be put into your project data analyses. And at CDD, we've really been thinking about FAIR and building our products in a fair respect for a while now, probably even before FAIR was came out with a paper in 2016. And we're not the only ones to do that, right? There's a lot of companies that have been thinking about how can I transform my data now that FAIR is becoming the big buzzword, right? How can I make my data fair? And I think that's one of the places where places like the Pistoia Alliance, they're really magical, right? Because it allows you to take all these, what would normally be competitive vendors or competitive companies, and bring them together in a non-competitive space so you can work on deriving standards or solution that can benefit the whole community. And so the DataFAIRy project was the brainchild of Isabella Fierberg, who used to be at AstraZeneca. And she was musing over this following problem. What I want is not what I have. So what I want is I want a lot of data that's really high quality and it's really well annotated. Where in the world could I get all of those? There's a lot of different places that have data. So there's public data and there's a ton of that, which is great, but it's not necessarily high quality. There's my data, which is perhaps well organized and annotated, but there's not a ton of it. And there's my partner's data, which may also be well organized, not a ton of that. But even if my data is well organized and my colleagues and my collaborators, even my competitors' data is well organized, if we're all organizing our data separately, we're creating a problem. We're not solving the problem. So if you just have a bunch of siloed organized data that don't interoperate with each other, you're at the same place that you started. And so that was the idea sort of behind DataFAIRy, is how can we come together as a community and build these data models or data standards to annotate bioassays that not only benefit the stakeholders, so the pharma companies that might be interested in sponsoring this project, but then be able to return that high quality annotated data back to the public. This is a problem. There's a lot of data. It could be costly. So what if we came together as a community in a pre-competitive space like the Pistoia Alliance to cost share the annotation? And the benefit for being involved in this as a pharma company is, one, you get to drive which assays should be focused on. You get access to that highly curated and structured data early, and then eventually is put back into the public sphere to benefit the whole community. And so a project team came together for DataFAIRy. It was 4 big pharma: Novartis, BMS, Roche, AstraZeneca. CDD and the Annotator was selected as the technology vendor. And this was all, like I said, made possible with the Pistoia Alliance. We heard some other talks from the Pistoia Alliance, I think yesterday, from Ernesto on the ontology alignment initiative. So they're a really great place. And if you're not members, I do encourage you to look into joining. But one of the things that sort of these stakeholders, those 4 pharma, came up with was a set of business questions that they wanted to define in moving forward with the DataFAIRy project. And this is really my soapbox, that it's the hill I'll die on. It's that you can't just create fair data for the sake of having fair data, or because it's the biggest buzzword, or because somebody's telling you to check a box. If you don't have a purpose to use that fair data, if you can't see the larger vision or understand how it's going to help you, it's not worth it. And it's not worth it because you're not going to be incentivized to use it. And I think that's one of the biggest problems that we face trying to get end users to try and adopt these FAIR standards, is understanding the benefit it could bring to them. And so, like good scientists who've listened to my soapbox spiel, have the same ideas, they wanted to come up with these business questions early on. So they had 26 initial questions that they wanted to be able to answer by having this high quality public bioassays annotated. They narrowed those down to 5 categories of questions that comprise about 15 separate questions. And it was things like around the biology. So a question like, I want to find a new, I have a new HDAC inhibitor project, and I want to find all the papers that describe the functional assays for measuring inhibition and use reporter gene assays, but I don't want them to be luciferase-based. Or they looked at assay technology oriented questions like, I have artifact assays for ADP glow kits been set up in the public domain, and what are the difference between those setups? They looked at chemistry and tool compound questions. So I need tool compounds for my assay, and so I need to find published results where X is measured, and I need to know the protocols behind them. They looked at specific assay conditions, like give me in vivo clearance data that was run in canines. And they looked at a computational aspect too. So I want to build a training data set for a virtual screening target for X. And because the public literature it's so abundant, if we're able to organize this data and ask a question of that organized data, like help me predict kinase selectivity, and we can probe not only our data, but our collaborators' data and the public sphere, we can build really good models. We can use those models, put the information back into those models, and really create a powerful iterative process. So this pilot project for DataFAIRy was really just that. It was a pilot. It was a kickoff to see what the larger project might look like, to learn some lessons so we could be better prepared moving forward. So at the start of this project, we really focused on just 500 public domain assays, which the goal was to convert into a FAIR format using an agreed upon data model, and that would answer the defined business questions. And we tried to select assays that were of different types. So assays that were formatted in different ways, had different levels of annotation. So we looked at commercial panels that were provided in a PDF, more semi-structured type format. We looked at PubChem NCATS assays, which are really large data sets that were pretty well annotated. And then we looked at assays from the literature that were originally in Kemble, where a target was one specific target, EGFR, and the reference was open access. And then we reselected 100 of these assays after they had run through the annotator annotation process to really stringent review by humans to really make sure they're high quality and that we would be doing a very good job putting them back into the public sphere. And one of the reasons annotator was selected as this technology is because it never took that human out of the loop. So there's some technologies that will do, wide scale markup of text, and it's great for that purpose for indexing, but it's not necessarily high quality. It can't necessarily make inferences from the text. So the fact that our annotator always had that human in the loop to check for any issues and allow an optional iterative QC process was really attractive to this project. So how well did the assays in the pilot get annotated? It really varied by field. So some did really well. So some fields were well annotated, but some fields were horrendously annotated. Things like controls or concentrations were missing. Things like target using any sort of identifier, whether it's uniprot or something from the protein ontology or DTO, some of those details were missing. But I think the most egregious error that I saw and really heightens the need for FAIR and interoperability is that oftentimes a commercial assay panel would be referenced. And this assay panel would no longer exist. It would have been bought out by another company, changed its name, or the website didn't even have a real URL anymore. You would go to try to find out more information about this assay that was performed per manufacturer protocol or to manufacturer standards, and find that you had no details on how the manufacturer or the CRO was performing it. And you would see those assays get propagated in references across papers. And suddenly you had a problem where you don't actually know how that experiment was performed. And so you can't use that public data in your model. So our main learnings from this were that you have to choose your assays wisely in a project like this. If you're walking into a set of annotations and they're all from the public literature and they are very old assays, one-off, and there's references that are to dead links or papers that are behind a paywall, there's a really high cost of doing that annotation because you have to sit down with the supplementary material, you have to go through it, it requires a domain expert, and it's not necessarily worth the cost benefit. Like I said, the commercial assay panels are going to be a really low hanging fruit for us to annotate. There's something that almost all pharma uses. They have detailed methods written up somewhere, not necessarily in a FAIR accessible format, but it's something that could benefit the community. Persistent links for commercial assay panel protocols. In my dream world, just like NCBI collects details for biosamples that are run, or SAR data, or GEO data. It'd be great if there was a collection of this is how the assay was performed. Or this was the kit that was used, and these are the parameters that are generally used with this specific kit or this commercial assay panel. You know, I know we're short on time, so just to go through a few other of these is that fully automated is not always fully accurate. So again, really heightening the importance of having quality metadata. So we have that annotator that can very quickly speed the human curation by using NLP and machine learning to suggest annotations. And we found that about 12% of the time, those annotations were wrong. For more difficult papers, that number increased. So having the ability to have a human take a step back, check to make sure the machine got it right, and then feed that knowledge back in the machine for training was something that was really important to us. And then finally, going back to sort of the ontology bridge, it's been mentioned a few times, there's really a need for flexible and standardized ontologies. The people we were dealing with in this project, they were really excited about F.A.I.R. They had some familiarity with it, but none of them are ontologists. And we had a number of ontologies that we used during the project, BAO, CLO, DTO, PR, PRO, units ontology. And we found, even so, there were about 200 terms that we had to request via the ontology bridge system because they just weren't represented in the ontology. And then you get to the question of, does that create any splits if we're requesting these ontologies, terms that maybe are in just other ontologies we don't know about? Is there a better way to set equivalents or link these things? So, you know, moving on to the next steps is that we consider this project a success. We annotated the assays. They're being re-uploaded into PubChem for the public with all those ontology terms linked to the assay metadata, but we want to scale this. So 500 assays in the scheme of things is not a lot. It needs to be something like 50,000. So we need to have more partners, which is going to lower the cost per partner per assay, and it's also going to lower the overhead cost. We need to optimize the assays that we choose. So we don't want to spend a lot of time on those assays that aren't widely beneficial to people. So a one-off assay in an obscure paper probably shouldn't get weighted as much as, say, a commercial panel that gets repeatedly used. And it's really important that we define the community standards and we align with vendors and publishers and government agencies. You know, we were a small team. We had our own biases on how we wanted the data presented. But if we get more partners, we get more brains, we get more input on the best way to structure and represent this data. So with that, I just want to thank the entire DataFAIRy team. They were in the previous slide, but there's also a lot of other people and different organizations that help make the project successful. So everybody at AZ, BMS, CDD, PubChem, Kemble, every other place on this acknowledgement slide that really played a part in getting this project to completion. Yeah, so with that, I will take any questions.
Thank you, Samantha, for that very in-depth talk in a very short amount of time, honestly. So the first question I have is from Hyundai, and it's, do you have any idea which assays for DataFAIRy would be the highest impact?
Yeah, definitely the commercial panels. I know that there's some interest from commercial vendors in standardizing their panels. It's beneficial for the vendors. It'd be hugely beneficial for data scientists who want to do data aggregation later on the results from these. And it's also really beneficial for the scientists who, instead of writing out the steps related to a particular assay panel, can just say, oh, I used this identifier. And if you go there, it takes you to a page that has all the details laid out nicely, ontology linked, in a standard data model for how that assay panel was run.
Going slightly off of that, how do you suspect to deal with the assay panels that you were talking about that the company has disappeared?
Yeah, so we're talking to a place, you may know them, BioAssay Ontology. So we've been considering adding a branch to BIO that would cover assay panels. And we would include in there the deprecated panels. So Ambit is one that I know ultimately was bought by Eurofins, but it's unclear to me whether they were performed the exact same way. But at least having that legacy data to say, okay, this panel is, if somebody refers to this Ambit panel in one paper and the other paper, at least I know that they're talking about the same panel. So the best we can do for some of these panels that we can't find the information for is just reference them appropriately. And the best case scenario, we can get a hold of the people that might actually have those data sheets somewhere in their labs and their filing cabinets and can provide us with the details to do a really thorough annotation.
All right. I think we have time for just one more question before we have to get into the breakout rooms. We're already a little bit over, but Lynn asked, for publishers, are you discussing any policy development?
This is my dream. So I firmly believe that the onus for this needs to be a burden on the publishers or the vendors or people sort of at the top of the chain. Why? Because if you tell a scientist they have to do more work, and I can say this because I am a scientist, I will not do it. But if the publisher requires me to have that in order to put it into the publication, then it's a good way to push that forward. A lot of the publications that we looked at that reference these assays that had no details about them, they were just allowed to be published. And that's because it was, it was fine at the time. FAIR wasn't a big thing. We didn't understand the implications. But to put this into practice on a community-wide standard, we need to get publishers and vendors involved. And I know that there are some really excited ones out there, some probably in this conference that are working towards this common goal.